Good day, everyone. Welcome to ALS Malaysia webinar series on COVID-19. In this video, we are going to talk about wastewater surveillance for COVID-19. ALS stands for Australian Laboratory Services, and its history started back in 1863, originated from Brisbane, Australia. We have presence in more than 65 countries with 400 plus laboratories worldwide, employing more than 13,000 people. In Malaysia, the management is headed by our General Manager, Dr. Chin Tin Tin, with a total staff strength of about 200 people. We have a total of 4 laboratories in Malaysia, with the HQ in Bukit Jelutong, Shah Alam, Selangor. Up north, we have a lab in Penang, down south in Johor Bahru, and East Malaysia in Kota Kinabalu. All our laboratories are also MS-ISO 17025 accredited for food chemistry, microbiology, and environmental testing. We also have 2 site offices at Kemaman and Ipoh. ALS Malaysia has garnered several recognitions over the years as a certified and MS-ISO 17025 laboratory. We are the recognized panel laboratory for National Pharmaceutical Regulatory Agency, Ministry of Health in Food Safety Testing, Jakim's Halal Panel Laboratory, Ministry of Environment and Water, Department of Fishery, Certified GMP Plus Laboratory for Heavy Metal, Mycotoxins, and Pesticides Testing. We are also the BioNexus Partner Laboratory for Bioeconomy Malaysia, providing testing services for all the BioNexus companies in Malaysia. ALS Malaysia was also awarded the IKM President Laboratory Award year 2017 to 2020 in the category of Best Commercial Testing Lab. In this presentation, I'm going to cover these few topics, which include wastewater surveillance for COVID-19. How could wastewater surveillance complement clinical testing? What has been done? And how is it done? Wastewater, also known as sewage, includes water from household or building use. For example, toilets, showers, and sinks that can contain human fecal waste, as well as water from non-household sources like rainwater and those from industrial use. Early this year, 2 independent studies in Netherlands conducted by Netherlands National Institute for Public Health and the Environment, RIVM, and the KWR Water Research Institute detected fragments of SARS-CoV-2 genetic materials in sewage system. And from then on, we see a lot more publications on detection of SARS-CoV-2 in wastewater. And there are also initiatives to start wastewater surveillance from government agencies worldwide. So what exactly does this signify? First, we need to understand that, as mentioned in the previous slide, wastewater is a mixture of many things. And human excrements, which are the likely source of viral genetic materials, are just a small part. When these fragments, likely degraded, in the presence of so much interference, are still detectable, it means that a considerable amount of viral particles must be present in the sewage system. And it would take a considerable number of infected individuals to shed this much of virus. So where are these viral particles from? How do they get into the sewage system? We all know that COVID-19 is a respiratory disease. However, many studies to date have revealed that SARS-CoV-2 can be present in the stool, and both genetic materials and live culture have been recovered from stool samples. It is understandable that the next question will be, are they still infectious? Is it safe to use a public toilet? How safe it is to work at a sewage treatment plant? All we know today is that viruses can still be viable when they are being excreted with the stool, and that the receptor required by SARS-CoV-2 to start an infection is present in the gastrointestinal tract, suggesting the possibility of gastrointestinal infection. However, to date, there is no solid evidence to show that transmission through wastewater is possible. Transmission through wastewater is not much of a worry for two reasons. First, the wastewater environment is harsh, and virus does not survive long after it leaves the host. Second, we simply will not let wastewater to get into our gastrointestinal system. However, unusual things did happen when all the most unlikely conditions meet. One well-known example is the Amoy Gardens superspreading incident of SARS-CoV-1 that happened in 2003. In this incident, an infected individual defecated. The aerosolized viral particles traveled upwards in the sewer pipelines and got drawn out from a dried up U-trap in the toilet at the upper floor. And hence, the virus spread in the apartment blocks, causing 342 infections and 42 deaths. SARS-CoV-1 and SARS-CoV-2 are closely related, and the size of the virus is certainly small enough to get airborne. However, the infectivity of these airborne viral particles has not been proven. There is a recent publication on a study trying to investigate a possible Amoy Gardens equivalent of COVID-19, in which the upper floor residents of the index patient got infected without having any contact. The authors suspected airborne transmission through wastewater based on the fact that, first, the three infected families have never come into contact, so it leads to the suspicion of transmission through touch surfaces. But when they tested the environmental samples, there is no trace of the virus in all samples collected from the public areas. Furthermore, other residents who had shared elevator with the index patient were not infected. Among all tested environmental samples, the concentration of virus is the highest in the bathroom of the index patient, which led to the belief that fecal aerosol could be the culprit. However, one limitation of the study is that there is no way to find out whether the U-trap was dried out during the occurrence. Still, at present, report on transmission through wastewater is still extremely rare. One of the biggest challenges in COVID-19 testing is the limitation of resources, both in the form of test kits and also manpower. To stop the spread of an infectious disease like COVID-19, ideally we would want to isolate all the individuals who carry virus until they get well. With an unusually high prevalence of asymptomatic and pre-symptomatic cases, testing seems to be the only way to find out who carry the virus. However, testing every single person is obviously not feasible in any setting. Using Malaysia as an example, our testing capacity is only almost 30,000 per day at its max. Even at a daily 30,000 capacity, it would take nearly 3 years to test our 32 million plus population. Even for Germany, with a much higher testing capacity, it would still take 3 months, and we are looking at a highly infectious virus. The research paper did a bit of calculation on how monitoring COVID-19 through wastewater can be a time-saving and cost-effective alternative, and it would allow routine monitoring over a prolonged period. Wastewater-based epidemiology, or WBE, is a surveillance method of monitoring public health through wastewater, which involves detecting and quantifying biomarkers present in the wastewater. In the case of COVID-19, when there are some unknown virus carriers among the residents of a city or a town, the viral particles shed in their feces will be collected at a local wastewater treatment plant, and this will act as a pooled sample of the whole community. So here is a brief outline on how it can be done. Samples could be collected routinely from wastewater treatment plants. Once an increased viral load is detected, the authorities will be alerted and targeted testing on the said area will begin. WBE is not a new idea. In fact, it has been used to monitor viral diseases before, such as polio, norovirus, hepatitis A, and influenza A. Besides diseases, it can also be tested to monitor alcohol and tobacco consumption of a targeted area by detecting the metabolites of these two substances, as well as to monitor the drug use over seasons by detecting the residues of the drugs in wastewater. To date, many studies have been done worldwide. Here is a table summarizing 14 studies carried out in 10 countries, including India, Australia, the Netherlands, USA, France, Italy, China, Israel, Spain, and Turkey, using the same method, following slightly different protocols, and detecting viral gene fragments ranging from 1.9 to 100 million copies in a liter of wastewater. Government agencies and city councils in the same countries have also started, in some countries have also started their local initiatives on wastewater surveillance of COVID-19, including the city of Tempe, New York, Utah in the US, NEA of Singapore, and also the Netherlands. Data from 7 locations in the city of Tempe can be found on their city council website. So how is it done? Here is a general workflow of COVID-19 wastewater surveillance. It will start from sample collection, then the sample will be transported to the lab and undergo a step that concentrated the wastewater sample to a smaller volume. Next, the genetic materials of SARS-CoV-2, a molecule called RNA, will be extracted. Detection or quantification of SARS-CoV-2 genes will be done using a technique called RT-qPCR. Lastly, the output will be analyzed accordingly. We'll look at the steps one by one. In the currently available studies, it is most common to collect 24-hour composite sample, whereby small portions of samples are taken over a certain interval throughout the day, and then pooled together. Another way is to collect the sample just at one time, and this is called a grab sample. The transportation of the samples can be done in refrigeration, or another simpler option is to keep it on ice along the transportation. It is always preferable to process the samples right away when they arrive at the lab. If immediate processing is not possible, then samples can be stored at 4 degrees Celsius for up to 2 weeks to avoid excessive freeze-thaw cycles. For storage above 2 weeks, it is best to keep at -80 degrees Celsius. -40 or -20 are okay too if -80 is not an option. Like I mentioned previously, viral particles in wastewater are often very diluted, and molecular testing methods often require a very small input volume. Therefore, a concentration step is often required. These are some of the methods that have been used for SARS-CoV-2 concentration, which include filtration through an electronegative membrane, centrifugal ultrafiltration, PEG precipitation, and high-speed centrifugation. The concentration step can increase the chance of detecting the virus should it be present, but it also increases the concentration of the matters that could interfere with the assay. So an optimal input volume would be a balance between both. Before proceeding to the actual testing step, an RNA extraction and purification step will be required to separate the viral genetic materials from the whole viral structure and also the impurities in the wastewater. It can be done in various ways. This diagram shows a general workflow of column-based method. To detect SARS-CoV-2 virus, the most common method is to target the genetic materials, which is in the form of RNA. The genome, which is the whole set of genetic materials of SARS-CoV-2, is around 30,000 bases in length, segmented into smaller units of genes that code for different functions. The targets for detection are very short regions within the genes, usually less than 200 bases. In the red box are the common genes that are used as a target. Before we go into RT-qPCR, we need to talk about PCR, polymerase chain reaction. It is a process of making many copies of short DNA fragments by enzymatic reaction through a series of heating and cooling cycles until it can be visualized in some ways. Conventionally, it is visualized as a glowing band on an agarose gel, sorted according to sizes. RT-qPCR is short for reverse transcription quantitative polymerase chain reaction, which is what we know about PCR plus an initial reverse transcription step that transcribes the RNA into its complementary DNA, which the enzyme can act on. Then it will move on to the step of making multiple copies. The visualization of RT-qPCR is through capturing the fluorescence signals emitted in every cycle, which reflects the number of copies being made in the reactions. PCR runs in cycles, and with every cycle, the copies of DNA double. In qPCR, the intensity of fluorescence signals increase along with the DNA copies. A cycle threshold, or CT value, is the number of cycles required for the fluorescence signal to exceed the background level. The more target DNA is in the starting materials, the less cycle it takes to cross the threshold. The number of copies of this target can be determined by plotting the CD values against a standard curve generated with the PCR target of known copy number. Each copy of these fragments would be equivalent to one viral particle. Here are some of the applications of COVID-19 wastewater surveillance. It can be an alarm for early warning. Several studies have demonstrated that SARS-CoV-2 has been detected in wastewater way earlier than the cases in the city got widespread. So in areas with no reported cases or gotten under control, a routine surveillance on wastewater could be performed instead of testing human. Wastewater testing can also be used as a pool testing strategy to monitor a high-risk community, such as prisons, dormitories, old folks' home, and also children daycare centers. By using a pool strategy, the number of required the number of tests required for a community is much reduced, and hence would be useful to countries facing shortage in testing resources. Furthermore, wastewater could be used for monitoring the progress of a mitigation plan implemented by the authorities, such as demonstrated in this France study, which show a correlation between number of active patients and the amount of viral load in wastewater. To date, there are still a lot of unsolved problems in how to properly incorporate wastewater surveillance into the COVID-19 surveillance framework, including there is no known standard of alarming level of SARS-CoV-2 genome copy. Virus spreading varies among individuals. Holding time in pipelines, diluting effect of rainwater, mobility of the residents, low sensitivity for large population, and interference by PCR inhibitors. And a take-home message for this session is that wastewater and clinical testing both have limitations and should work hand in hand to tackle COVID-19 crisis. So before ending this presentation, I would like to show you some of the key testing services provided by ALS. Our environmental testing services included water services, testing for soil, sediment, and solid waste, air quality monitoring, other specialized testing services, and microbiological services. Our industrial hygiene testing services included analytical services and occupational health medical surveillance program. Our food safety testing services include microbiological testing, chemical and nutritional labeling, contaminants, shelf life studies, and halal testing. Our biotechnology testing services cover DNA testing for both food and environmental samples, and we also provide services for medical devices and pharmaceutical testing. Last but not least, here's our list of tribological testing services. Thank you for your time. Should you have any question for us, feel free to contact us to discuss about your testing needs. Have a good day.